welcome to Quantum Photonics. Anxiety disorders affect millions worldwide, but what role does our gene play? To answer this, a groundbreaking study analyzed data from over about 1.2 million participants spanning five major continental groups. Among them, nearly 100,000 individuals had anxiety disorders. This massive effort identified 51 specific genetic regions linked to anxiety, 39 of which were brand new discoveries. What's fascinating is how these findings crossed ancestral lines. Genetic risk factors identified in people of European descent were also seen in African, admixed American, East Asian groups, showing that anxiety's genetic roots transcend geographic and ethnic boundaries. The researchers also found that anxiety has deep ties to the brain, with many of the genes linked to areas like the limbic system, the cerebral cortex, and brainstem, regions known for their role in emotion and stress regulation. By looking even deeper into how genes work in the body, the study uncovered 115 genes tied to anxiety through processes that involve both the brain and the other tissues. Interestingly, anxiety doesn't exist in isolation. It shares genetic links with mental health conditions like depression, schizophrenia, and bipolar disorder. Moreover, these connections extend beyond mental health, showing overlap in various physical health conditions. This study underscores the importance of studying diverse populations and using advanced tools to understand the complex web of factors behind anxiety. It offers hope for more personalized approaches to treating anxiety disorders in the future, benefiting people from all walks of life. So this very interesting and detailed study was done by the lab of the Poliman team, the lab of our guest speaker, Dr. Renato Polimanti, who is an associate professor of psychiatry and of biomedical informatics and data science at the Yale School of Medicine and an associate professor of chronic disease epidemiology at the Yale School of Public Health. He is also the Associate Director for Scientific and Technical Operations of the Clinical Epidemiology Research Center at the Veterans Affairs Connecticut Healthcare System. Dr. Polimanti's primary research interest is to apply big data analytics to molecular, clinical, and imaging data to understand the biology and the epidemiology of human traits and disease and to develop tools to improve the healthcare of diverse population groups. So without further ado, I proudly bring to you our guest speaker, Dr. Renato Polimanti. Renato, welcome to Quantum Photonics.
Thank you so much for the invite. Like, it is a pleasure to be here. Again, thank you so much for the introduction and for the nice summary of the study. I'm very happy to talk about our research and about the study that we conducted. So I know that you talk about a broad range of scientific topics, and so my presentation is going to give like some basic knowledge about genetics and about psychiatry, and then talking about like the specific of our study. I sent Cecile like a slide deck with some figures that we can go through together, and so like I will have like some graphical help to show you what I'm talking about. And so like if you downloaded the PDF, we can start with the first slide, which is the title of our study. Again, my name and my contact. Again, I will show them also at the end. And so like we can start our chat together. Going to the second slide, which is my competing interest. I receive an honorarium from Karger Publisher for my work with them, and also I received in the past a research grant from Alkermes. Both of these competing interests are not related to the work that I'm showing today, but anyhow, I wanted to mention them. So if we go on the third slide, this is a picture of my group at Yale. And again, like we are in the Department of Psychiatry, and our primary interest is biological psychiatry, trying to understand which are the biological processes that are predisposing certain individuals to develop psychiatric disorder, symptoms, but also behavioral phenotypes and other comorbid conditions. We do that using human genetics, molecular epidemiology, computational biology. I started as an evolutionary biology when I got my PhD in Italy, and so that's also a thing that I'm interested. I'm not going to talk about it today, but one thing that we try to do is to connect biology, psychiatry, and human evolution. Here is also the link to my lab if you want to learn more about what we are doing, our research. Take a look, and again, there are also other studies that we conducted about different psychiatric disorders. And then if we can go on the fourth slide, we can see the title again of the study I'm going to talk about, the list of the people that contributed. These are current or former member of my lab, and again, they are amazing group of people that provide a different contribution to this study. I also put a QR code if you want to scan and access directly to the web page where the article is available. And then, like, if you go on the slide 5, there is the picture of two people. One is myself. The other is Eleni Friligo. She was the leading analyst for this project. She was a postdoc in my group, and again, she's an MD. And so, like, now she started a residency at Yale to become like a psychiatrist, because her goal is to become a physician scientist. And here I put the link of our press release where we give like a summary of the study and a few key points. Again, you can also feel free to read our press release. And so let's go to slide 6 and let's start to talk about anxiety and genetics. So here we see two graphs. On the left, we have the prevalence of different psychiatric disorders. We have anxiety, MDD is depression, ADHD, people are very familiar with this abbreviation, PTSD the same. Then we also have BPD, which is bipolar disorder, eating disorder, OCD, autism, schizophrenia, Tourette syndrome. And so we can see that anxiety, if we include all the different anxiety disorders, we are going to talk in a little bit about different definitions. We see that about between 25 and 30% of the population during their lifetime, they will develop like the disease. This is like a massive prevalence, the highest among all psychiatric disorders. And then on the right, we can see another information about the irritability. What is the irritability? Irritability is the genetic component that explain the cases. So about 40% of the predisposition to anxiety is due to genetic factors. And we can see that there are differences. There are, for example, other psychiatric disorders that have a higher irritability, like schizophrenia, ADHD, Tourette syndrome. There are others that have lower irritability, like depression. Again, there are different ways to calculate irritability. The one that I'm showing here is based on twin studies, so on family information. And so, like, again, also there are different ways to calculate. And for anxiety, we have a lot of heterogeneity and a lot of different kind of diagnostic definitions. And so, like, it's not that we are 100% sure that this number is around 50%, 40%. And there are still studies ongoing to estimate the amount of irritability that can help us to understand the predisposition to anxiety. And then let's talk about what irritability means. And so if we go on slide 7, I'm reporting here schizophrenia as an example to understand what irritability means. So I say that anxiety has around 40-50% irritability, schizophrenia 80%. What this means? It means that people that are related to each other, if one of these person is a case, the person that is related to that has an increased risk. So for example, monozygotic twins, which are the identical twins, if one twin has schizophrenia, the other twin has 48% lifetime risk of the disease. Dizygotic twins, so the non-identical twins, or like also, again, like that are similar to siblings, they have around 70%. A child of a person that has schizophrenia, 13%. 9% siblings, 6% parents. then we have half sibling, and you see the less genome is shared between two individuals, the less risk we have. So the general population risk that we have is about 1%. And so like this, what it means heritability. So people that are related to each other share pieces of the genome, and sharing pieces of the genome, they may have increased risk in the family. This is why certain psychiatric disorders tend to occur in certain families, but also other diseases like cardiovascular disease, cancer. There are certain families where this disease occurs the most. And this is because in those families, we have certain regions that get inherited together, and they have some variants in the genome. And so like this is what is predisposing people to develop certain diseases. And now if we go on the slide 8, we can see few numbers about how complex is the human genome. So the human genome is kind of a chain, and the unit of this chain are nucleotide, which has small bricks that build the human genome. In a single human genome, there are more than 3 billion base pairs. Currently, we know that there are more than 300 million known variants. So in the human genome, there are positions that don't change, and there are other positions in the genome that change. And we know that there are more than 300 million positions in the genome that change. But not all these 300 millions happen in every individual. Each of us approximately carry between 4 and 5 million variants that we inherited from our parents in most of the cases. And then we have different type of variants. The vast majority of variants are single nucleotide variants, SNVs, or short indels, which are short insertion or deletion. So a single nucleotide variant, if you look on the right, on top, you have a single nucleotide that change. So we have 4 nucleotides, which are the letters that build our genome. So we have adenine, which is known with A, C, which is cytosine, G is guanine, and T is thymine. And so like these are the 4 bricks that build the human genome. And so like in a certain position, people may have a different nucleotide, and so we have a single nucleotide variant. In another position, we may have like a nucleotide that is missing in a person, and so we have a deletion. Or we may have an additional nucleotide, which we have an insertion. And then we can have also more complex, where certain regions tend to flip or duplicate, and so things can get very complicated. But the vast majority of the variants that each of us have are single nucleotide variants or short insertion deletion. And so why I'm talking about this? Because these are the variation that is predisposing people both to develop physiological traits like being of a certain height or having a certain skin color, a certain eye color, or to develop certain disease. So the variation that we have, which is again, most of the cases a good thing, in some cases develops people to develop disease. And then if we go on slide 9, we can start to talk about how the human genome is built. And so like here, there is a first concept that we need to discuss. The first is about what the human genome does. The primary function of the human genome is to code for proteins. So there are certain regions in the genome, which we call genes, that code proteins. And proteins are the basic, the fundamental unit that build biological systems in humans, but in every living organism. But if we look at the human genome, only 2% is coding DNA, which are regions that are coded into proteins. 98% is non-coding DNA. And so like, what is this non-coding RNA? This non-coding DNA is DNA that doesn't get coded in proteins, but does something else that we are going to talk about. And so we have coding regions that get translated into proteins and non-coding regions that don't get translated but do other things. Let's talk about the coding regions. And so here we have our cell, and we have our, if we go on the slide 10, we have our nucleus, and so this is like the kind of a representation of a cell. And in nucleus, we have our genome. So most of the human genome is in the nucleus. And then like we have, as I mentioned, coding regions, which we call genes. And in genes, there are exons and introns. And these get transcribed in RNA that get spliced in messenger RNA, mRNA, then get transported outside the nucleus, and that gets translated into proteins. And with the proteins, then all biological systems are built. Indeed, what proteins do, if we go on slide 11, proteins are needed for building the immune system, to build muscles, to building all the structures that are in the human organs, to be present in cell membranes, to act as enzymes, are receptors. They are enzymes that we need in our digestive systems. They are in the blood. They are the one that, like hemoglobin, they transport oxygens. Again, and there are also immune system cytokines that are able to communicate with other cells. So proteins are extremely important. So primary goal of the genome is to build the proteins. But we saw that only 2% of the human genome is coding protein. So 98% is not coding protein. So this 98% is important or not. And we can check and compare with other organisms. And so here, if we go on slide 12, we have different organisms with a very simple one, like prokaryotes, one-celled eukaryotes, fungi, plants. We see that we are seeing here, like on this slide, on the y-axis, the percentage of the non-coding genome. And so we see that very simple organisms have a lot of coding. In proportion, they have a lot of coding genome and very little of non-coding. The more complex it becomes the organism, the more increased the non-coding genome. And so what this means? Maybe it means that the more complex is an organism with multiple biological systems, multiple cells, multiple tissues, multiple organs, the more we need the non-coding region. And if we go on slide 13, we see that the non-coding regions have non-coding elements that regulates the genome. So that's why they are important. So here we can see, again, our chromosomes, so the X structure, and then we have this chain, which is our DNA, which is wrapped around certain proteins that are called histones. And so and then we have again our DNA, and then we have different elements. As I mentioned, we have the genes. We have elements outside the genes that regulates. We have an answer, which is another non-coding regulatory element. And so all these things, all these non-coding elements are regulating the expression of the genes in different tissues, in different cells. And so they are important because they tell the DNA where they need to generate proteins. And so this is why more complex organisms have more non-coding genome. And so this is also a challenge because, again, like all these non-coding elements can regulate multiple genes. And so in these elements, we have genetic variants that we are studying and we are identifying, like the study that I'm going to talk about. And so the goal is to find which are the variant and then to understand what they do. And so this is what we are going to talk about today. And so in slide 14, let's start to discuss about how disease or human traits gets inherited. There are different kinds of inheritance models. And so here on slide 14, we can see two, the monogenic inheritance and polygenic inheritance. There are others, but again, these are kind of the two extremes that I want to show you. Monogenic inheritance, in this case, in this figure on the left, we are seeing an autosomal dominant transmission. Autosomal dominant, so in our, we have in each cell of our body 2 copies of our DNA. So it means that we have twice the number of alleles, of variants. And so here we have a father that has a disease, and this is also carrier of a mutation that is associated with this disease. And so we have this light blue variant, and then the dark blue is the normal nucleotide. And so like we see that the mother is not affected and is not carrying any disease, any mutation. And so like these parents have the children, have 4 children, and we can see that 2 of the children get the mutation, and so they are also affected. And so we have that if they have 1 copy, they are affected. And so we have this thing that if the mutation gets transmitted, also the disease gets transmitted. So the mutation is the cause of the disease. And this is the case for several rare disease where we have a single mutation that usually is very rare in the population, and if like the parent transmits the mutation, the child has the disease. However, this is the transmission mostly for rare disease. Most of the common disease don't follow this transmission. They follow a polygenic inheritance. And so on the right, we can see our chromosomes. So we have 24 chromosomes. So 22 are the autosomes, so are the non-sex chromosome. And then we have an X and a Y. So in this case, we have a male because it has both X and Y. And in this chromosome, we have multiple variants that here are indicated with B. And here you can see that there are 2 types of B, orange Bs and gray Bs. So in this case, the orange Bs means that the variant is associated and increase the risk of a disease. And so this person has multiple Bs, orange Bs, and some gray Bs. If we add the effect of each of these orange variant, we will have a certain number. And so we will be able to assign a certain risk. So the more orange B we have, the higher is the risk. So if we look on the bottom right, we will see that there are some people that have a very high genetic risk. This is because they have many of these orange variants. Then in the middle, we have most of the population that don't have a lot of them, but they have some. And then on the right, we have few people that have very few of these orange variants. And so they have a very low risk. And this is the mechanism how most of the disease, including psychiatric disorders, including anxiety disorders, get inherited. There are these variants that everyone here has, in some cases, a very high number of anxiety-associated variants, in some cases, very low number. And so in the population, there will be some people that have a very high genetic risk, and they will develop the disease. And now let's move on to the next slide, which is to explain about which variants are more important. Again, here I'm showing an example about schizophrenia, because again, like it's a well-studied disease both for rare and common variants. So what I'm talking about here about rare and common. So here we have a graph where we have the allele frequency, so the frequency of the mutation in the general population. So we have some rare mutation that are associated. Odds ratio is an estimate of how strong is the effect of a variant with respect to a disease. And so like the higher is the number, the strongest is the effect. And so here we can see there are some variants that have a very large effect on schizophrenia, but they are very rare. So only very few individuals will have these variants. And then on the right, we have a lot of variants that tend to have a much higher frequency, so they are common in the population. Most of us will have few of these variants, and the effect is very small. So none of these variants by themselves can cause schizophrenia, but they can add a little bit the risk. So my question is which variants are going to be more important with respect to the disease? So again, if we look to the effect size, the more important variants could be the ones that are very rare, but they have very large effect. Because on the other side, there are variants that are common, but the effect is super tiny. You can see that it's very, very small. But if we look to another example, again, in this case is autism spectrum disorder, which is a disease that is well known to be affected by rare mutation. And so we can see here on this plot that there are different kind of rare mutations that are known to be associated with the autism spectrum disorder. But if we put all of them together, they explain all these rare variants, they explain very little, about 10% of the cases. But so what about all the others? So common variants, so the variants that we saw before that they were very small effects, but they were common, not alone, but combining their effect, for autism, they expect about 49% of the cases. So they are very important. Although the effect of each one of them is very small, the cumulative effect of all of them together is very important. And so like the variants that we are going to study in the article that I'm describing today is common variants related to anxiety. And so which kind of approach we are going to use to find this common variant? So we are going to use a genome-wide association study. So genome-wide means that we are studying the entire genome, investigating millions of variants. And so like what we are going to do is to compare individuals with the disease and individuals without the disease, and trying to understand if there are some variants that have a frequency that is higher in the cases, people with the disease, and has a lower frequency in the controls. So here we have three examples. SNP, which is another way to call single nucleotide variant, is a single nucleotide polymorphism. We call it polymorphism when it's common. So we have three examples about variants: SNP1, SNP2, and SNP3. So we can see that SNP1, we have which is the variant as like this green color. We have that the frequency is similar both in cases and controls. There are not many variants in both groups. And so we can say that there is no association to the disease. Then we have the SNP2, where we have a lot of these different variants in our case group, but a similar number also in the control group. And so also SNP2 is not associated to the disease. And then we have the SNP3, where we have many of these variants in the cases and very few in the controls. So we can say that SNP3 is associated with the disease. And so what we are going to do, we are going to do this kind of comparisons millions of times across the entire genome. So this study design is not based on a priori hypothesis. So we are not deciding which gene is associated with the disease, in this case anxiety. We are simply testing all the variants that we can across the genome with respect to disease. So we can call this as a brute force experiment. We are leveraging this massive sample size, more than 1.2 million participants, to find which are the variants associated with anxiety. So let's go on slide 18. And so like here we have kind of like a summary of all the GWAS conducted so far. So up to November 20 of this year, we have more than 7,000 GWAS articles. So articles describing genome-wide association studies. And here we have a plot. If you look closely, we can see these gray lines. And we can, if you count them, you can see that there are 23. So there are all the chromosomes here. So the gray line are the chromosome. And then there are these dots that are connected to each of these gray lines. Each of these is an association. So which means that we have all associations that are spread across the chromosome. It's not that there is one chromosome that has more association than others. And so like so far, we have almost 700,000 associations about different diseases. So this plot kind of is like, it looks like a puzzle because it is a puzzle. So what we are finding is all these pieces of different disease, and what we are trying to do is to understand how they are all connected. And so like, what are the key messages that we get from this plot? We get two main messages. First, most of human traits and disease are polygenic, which means that the variants that are associated with their predisposition is spread across the genome. They are not located all in a specific region. The second is a new term that I'm going to mention, which is pleiotropy. You can see that in different regions there are multiple colors. So it's not that in a specific region there are only psychiatric association. and in another region there are only metabolic association, and in another one there are only cardiovascular association. They are all mixed together, and this means pleiotropy. So each gene is associated with multiple traits, with multiple phenotypes, with multiple diseases. So this increases the complexity. Multiple genes can be associated with different disease, and we will talk about this in the concept of psychiatry, showing how anxiety is also connected with other psychiatric disorders, but also with other health outcomes. Let's move on to slide 19. So I talked about like these 1.2 million participants, and where did we find all these participants? We found them around the world. And so we used 6 large cohorts that were generated by different efforts, like the Psychiatric Genomic Consortium, which is an international collaboration of different scientists bringing together the samples, UK Biobank, which is a large biobank in the UK, FinGen, which is another biobank from Finland, the Million Veteran Program, which is a US biobank funded by the Veteran Affairs to investigate the genetics of veterans and help to improve their health care. As eyesight, which is a Danish biobank focusing on psychiatric research, and the ROAS research program, which is a US biobank but focusing on the general population. And so since we are in the US, I'm going to give a little bit more information about the 2 US biobanks that we use. One is the Million Veteran Program. This is a national research program funded by the Department of Veteran Affairs. And here you can see the map of the US, and you can see all the centers where the recruitment is still ongoing. And so the goal of this study is to generate new insights about molecular mechanisms that are at the base of disease that are affecting veterans. So opioid sensitivity, post-traumatic stress disorder, cancer, heart disease. And so like we were able to access the data from this study and combine it with others. The second is the All of Us research program. Again, we have another map of the US, and here are all the centers where people are being enrolled. And so also in this case, the goal is to do gene discovery and translate this information in instruments that can help people to live better and live longer and be healthier. Okay, so let's talk about how we were able to combine all these definitions. So if we go on slide 22, we have a table with the different 6 cohorts that we combine. And then we have like a population, because again, we have different population groups. And you can see that in the US, we were able to have more diversity, and in other groups, European groups, we had less diversity. But also the other issue was about the different diagnostic definition. So for example, we use different information. For some biobanks, like the All of Us research program, FinGen, Eyesight, and the UK Biobank, we used information from the electronic health records. And so like we use different codes related to different disorders, because again, anxiety disorder is a broad definition. And so we were able to combine them. Then for the Million Veteran Program, we use self-reported information about symptoms related to generalized anxiety disorder. And then the Psychiatric Genomic Consortium combined different definitions of anxiety disorder. So you can see there is a little bit of variability among all these definitions. So if we go on slide 23, we did an initial analysis to decide what was the best way to combine them. We did a genetic correlation analysis. So among all these cohorts that were assessed for different anxiety disorder definitions, we calculated the amount of genetic predisposition that was shared between these definitions. Again, they are all about anxiety, but there are differences. And so we can see that in some cases, we had a high genetic correlation between, for example, between the All of Us research program and the Psychiatric Genomic Consortium. The genetic correlation was basically 1. And then we had other issues where the genetic correlation wasn't that high. For example, between Secondary Genomic Consortium and ISAIC. For example, in this case, was due to probably the definitions, because ISAIC, they use a very strict definition of generalized anxiety disorder. In PGC, the Secondary Genomic Consortium, they use a very broad definition. And so because anxiety is very heterogeneous, that reduces the genetic concordance between these two definitions. So how we combine this information? If we go on slide 24, we use a method called genomic structural equation modeling. So we use the information about their genetic correlation to find a factor that was shared across all these definitions. And so we found this ANS, which we call anxiety factor, that was well connected with all the codes that we had. So we can see that there are some numbers here. I'm not going to explain what they mean, but you see that the arrow that connected the ANS factor to each court is higher than 0.8. And what this means, it means that there is a good connection. And so we did a genome-wide association study of this ANS factor, combining all the information that we have from these different courts. And again, another key point of our study, if we go on slide 25, is about the diversity of our study. So unfortunately, again, like this is a big issue, and I'll show you a couple of slides why this is a big issue in genetic research and also for psychiatric research. And so like we were able to access data from diverse population. For example, the All of Us research program, we had information about 5 population groups. In some European countries, like the Finnish one, the Danish one, we only have access to Europeans. In the Million Veteran program, we had information about European and African Americans, and so on. So we were able to combine them. Again, but if you look the numbers, which is the total sample size, you could see that European descent individuals were the vast majority. Again, and this is an issue not only for our study, but for most of human genetic study. And so if we go on slide 26, this is the problem that human genetic research has so far. So here we have a plot where we have years on the bottom, and then the number of individuals in millions included in genome-wide association study, not only in ours, not only in psychiatry, but all genome-wide association studies. And you can see that most of the studies are done in population of European descent. Things are improving in the last few years, like we are increasing, but we are still very far from representing the distribution of global population, which is the plot on the right. So you can see like how each population should be represented, and how we are currently over-representing European descent with respect to the other population groups. And what about psychiatry? So if we go on slide 27, in this table, you can see all the genome-wide association study that we have up to 2024. There is in red our anxiety study, which we have 5 population groups. But then there are still some studies that are conducted only in Europeans, or only in Europeans and African, or again, there are studies that are getting there. And so like you can see also the sample size. There are several studies here that have a sample size greater than 1 million. There are others that are not still there. And so like things are moving. Again, you can imagine that compared, for example, to 4 years ago, the sample size of this study is almost 10 times bigger. So things are progressing very, very quickly. But the goal right now is to not only increasing sample size, but also increasing representation. So, and representation, it's not only important in including diverse population, but also correcting, having a correcting, not only including them, but also having a correct way to analyze them. And so there is still a debate in genetic research, but there was this report from the National Academies of Science, Engineering, and Medicine about which are the population descriptors to use in genetics and in genomics research. And so like there are 2 kind of like approaches. One is using a race, ethnicity category, and one is using genetically defined, genetically inferred ancestry classification. And so the things that I want to show on the slide 29 is that how race, ethnicity are social constructs. They change over time. And so here we can see how in the US, the race and ethnicity classification changed over time. And so we can see here that they don't reflect biological concepts, but they reflect sociocultural concepts. And so like if we go on slide 30, we have instead like genetic diversity and what these a few key numbers to represent that. This is an old study from 20, we are in slide 30 now. We have like this study from 2008. They was one of the first genome-wide study to investigate human diversity. Here we have more than 50 population from different continental groups. And so like in this study, we can see that the majority of the diversity is between individuals within each population. So the majority of the diversity that we have is between two individuals, independently from where they are from. And so like this means that most of the variation that we see is only because two individuals are different from each other. For populations that are within the same geographical regions, we have very little variation, about 2%. And then from different continental groups, different geographical regions, the variation is about 9%. So again, like we have these differences, but again, most of the differences are between individuals. And then we have about 10% that are between regions. And this is a very important concept because again, like we need to understand that human species is not very diverse. Most of our diversity is between individuals, and there are 10%, around 10%, which is up between populations from different continental regions, which is not very high. And the other concept that we need to understand is that human genetic diversity is not different groups, but is a continuum. And so like all this, in this plot, which again, like is a representation of a biobank, which again has different kind of like distribution, which is not the distribution that we have in the world. But the things that I want to show you is that everything is connected. It's not that we have different boxes. It is kind of like continuum of diversity that then we label in a certain way to model this diversity and to avoid like possible artifacts in our stuff. So if we go on slide 32, there is kind of a summary that I brought together a collaborator in my department. She's working on racial discrimination, racial equality. I'm working in genetic research. And so we came together to try to distinguish the concept of race and ancestry. So racial groups are a group of individuals that may have similar physical appearance that are also mainly driven by cultural, social, and linguistic similarities. Ancestry, on the other side, are genetic similarity of people that share similar demographic history within the same continental groups. And how these are defined. So racial and ethnic classification change over time, and they change because of sociocultural context, and they can change across population. So for example, the word ethnicity in the US mainly reflect Hispanic and non-Hispanic individuals. In the most of the rest of the world, ethnicity means something else. It means like cultural differences in each population. And so you can see how the terms is different. Instead, how we define a genetically inferred ancestry, we use genetic information to try to understand biological similarity using reference panels. And so in research, when we should use race and ethnicity classification, we should use it when our goal is to understand racial discrimination, racial biases. If our goal is to understand social, cultural dynamics, so we should use race ethnicity. If our goal is to investigate biological mechanisms, we should use genetically inferred ancestry, and also try to use quantitative modeling to kind of model this continuum that we have in human genetic diversity. And so in our study, we use a reference population around the world. We are on slide 33 right now. And so these are from different projects. And so we were able to classify our individuals using this genetic information. So we used genetically inferred ancestry and not race ethnicity. And so now we're going to talk about our study. So we did a genome-wide association study. And so we were able to identify variants that were statistically associated with anxiety. And so if we look on, we are on slide 34, if we look on the plot at the center, we have this plot where there are numbers at the bottom, 1, 2, 3, up to 22, which are the autosomes, the chromosomes that are not sex chromosomes. And so then we have different dots, and some dots get very high. This means that variants that are associated with anxiety. So the higher is the dot, the strongest is the association. And then around this plot, we have different methods. And so in our study, we use different methods to understand which are the pathway, biological pathway that are associated with the disease, mechanism that link genetics to other molecular mechanism, for example, gene expression, which is also called transcriptomics, or also protein. We already talked about how important are proteins, and so we also use protein information to understand better biological mechanism underlying the association. So if we go on slide 35, there is a summary of what we found. So gene discovery across populations and omics domain. So because the sample size was larger in individuals of European descent, we had more than 40 genetic associations in this group. Only one association in Africans and none in the others, because the sample size was much smaller. But when we combine all the populations, we were able to find additional 10 genetic associations. And so in the plot on the right, we have again our chromosomes. Each dot is an association. On the one on the bottom, the one that we see genome-wide association study, each dot is a variant. The higher is the dot, the stronger is the association. So we have a total of 51 genetic associations. But then we also, as I mentioned, combined genetic information with other molecular data. And so when we combine genetic information with transcriptomic information, which reflect gene expression, we identified 152 associations that were related to the gene expression in the brain. And then 94 associations related to the gene expression in the tissue, and then 24 associations that were related to the proteomic expression in DLPFC is dorsolateral prefrontal cortex, which is a region in the brain. And so we use these different associations to conduct genome-wide association study, transcriptome-wide association study, and proteome-wide association study. So we have 3 layers of molecular information: genes, gene expression, and proteomics. And so if we check which variants are associated with these 3 layers and go on slide 36, we see that we have 6 genes that are significant across GWAS, the genome-wide association study, TWAS, transcriptome-wide association study, and PWAS, proteomic-wide association study. And so here we have names of these different genes, and we can see that these genes, in most of the cases, were previously implicated in brain biology or in brain disease for 5 of them. And so again, these are all mechanisms that we are currently trying to understand better. Again, how, for example, the first one, CTNND1, which is involved in spine and synapse development, how this is related to anxiety. The second one, which is a CNNM2, which again, these are all names of genes, is related to neuronal development and epilepsy. And so on, we are trying to understand how these different biological mechanisms that play a role in brain function are related to anxiety. But then on the bottom one, we see a gene that is not immediately related to brain biology, which is called KHK. So this gene encodes for a protein, which is an enzyme that metabolizes fructose. So again, this is sometimes what happens with this kind of like large scale study. We find something that we don't expect, and we need to follow up to understand better what this gene does. There are some studies that pointed out the possible impact of high fructose diet early in life can predispose to anxiety. These were done in animal models, and so this gene could be related to that mechanism. But again, like, there are many other studies that we need to do before we can make a conclusion. Then there are other genes that we found. On slide 37, there are two genes that were previously known to be related to brain disorders. For example, CNTN5, which was related to suicidal behaviors, autism. We found these genes also related to anxiety. And we know, for example, that anxiety and suicidal behavior can be comorbid. The other one, for example, furin, was previously associated with schizophrenia. And so like, and in the mechanism that we found is that the variant kind of increase anxiety because probably is reducing the expression of this gene, which plays a role in some biological mechanism that people are starting to understand right now. And so also in this case, we are finding something new about a gene that was known to play a role in schizophrenia, but probably plays a role also in other psychiatric disorders. And then we also find other things that were not very known. For example, in slide 38, we found a gene that doesn't code for a protein. So this is called a long intergenic non-protein coding RNA. And so as I mentioned, like 98% of the genome is non-coding. This gene is non-coding, doesn't code for a protein, but has a regulatory function. And so we found this to be the strongest association in individuals of European descent. But again, it doesn't code for a protein, so we want to understand better what it does. And also, like, it doesn't appear to be expressed, this RNA, in the brain. And so how is that connected? One possible mechanism, we saw that this specific long non-coding RNA is expressed in the testis. And so like how this is connected to anxiety, again, like we know that anxiety disorders is different between women and men. And so like we also have some evidence about possible hormonal regulation that play a role in different psychiatric disorders. And so like this long coding RNA may be related to this hormonal regulation, which may acting through the connection between testis and brain, which happens only in males. Could be a protective mechanism, could be a risk mechanism. So this is something that we need to understand better. Previously, the same non-coding RNA was associated with smoking initiation, externalizing behaviors like risk-taking behavior, and psychopathology in general. So again, we need to understand better what this does. If we go on slide 40, then we have like an analysis where we integrate the genetic information with gene expression in different brain regions, actually in 13 brain regions. And so with this analysis, we found 152 transcriptomic associations. So associations that are driven by the gene expression in the brain. In some cases, we found genes that were associated in multiple brain regions. In other cases, we found that the gene was associated with anxiety only because of its effect in one specific brain region. And this was the case for the strongest association, transcriptomic association that we found, which was the RD2, which is present only in the cerebellum. And so in our study, we found that the RD2, which encode for the D subtype of the dopamine receptor. So this gene is important because kind of like it regulates transport dopamine between the synapse. And so like here we have like a mechanism that is linking this gene to anxiety. This gene was previously identified also with respect to other psychiatric disorders. Again, schizophrenia is one of the disorders. And so like we need to understand better how this can work. So we are in slide 41, if I didn't mention it. There is a nice picture about the synapses and the transporter and the receptors. And then we talked about cross-population analysis. And so, as I mentioned, we found a lot of things in individuals of European descent because of the large sample size, but much less in the other because of the smaller sample size. So we can do a different analysis, which is called polygenic risk scoring. So we can combine the effect of different variants and try to stratify our cohort based on their genetic risk. And so, for example, here we divided our cohort in 5 groups depending on their anxiety genetic risk. And so people on the first group, slide 42, they have a risk that is 1. So we are going to consider them as the reference. In the second quantile, which is the people that have slightly higher genetic risk, we can see that the odds ratio, which is the factor that measure the statistic that measure the association, tend to increase and increase in all populations. And then in the third quartile, increase again, fourth quartile again, and the fifth quartile, we have the highest. Then we can see that each dot has a different color, and so like again, like is a different population. But then we have also like lines connected to that dot, which is the standard error. In some cases, we have larger standard error because the sample size for that population was small. And so here, for example, we can see that Europeans tend to have the smallest errors because we have the largest sample size, but also they tend to have the highest effect. What this means is that if we want to use polygenic risk scoring to stratify anxiety risk and identify which are the people that have higher risk, we need to have better representation. In this way, we will have better assessment as smaller standard error and like effects that can reflect association across different populations. As I mentioned, is about 10% the variability across human continental groups. But again, we still need to have that 10% well represented to be sure that if we are using information, we are going to provide the same quality of feedback for population independently from their backgrounds. And then on slide 43, we are going to see some data about shared statistics. So we talked about genetic correlation, which is the estimate of genetic variability, liability that is shared between 2 different outcomes. And so we have RG, which is genetic correlation, for example, between anxiety and major depressive disorders, anxiety and bipolar disorder, and anxiety and schizophrenia. So the genetic correlation between anxiety and major depressive disorder, it was 0.9. So which means it is very high. Anxiety and bipolar disorder, 48, and anxiety and schizophrenia, 0.45. So we can see that the genetic correlation between anxiety and depression is very high. And it also means this is in line with what we see. We see that people with symptoms of anxiety and depression tend to occur together. And then on the circles above that, we have the number of variants that are shared between them in thousands. So again, we are estimating that there are about 11, 12,000 of variants that are associated with anxiety. It's not the one that we found, but is the one that we are predicting to be present. And 11 of these 12,000 are shared between anxiety and depression. So they are kind of like variants. There are a lot of variants that affect both. And then there are variants that are shared between anxiety and bipolar, which is around 8,000, a little bit less than 8,000. And then variants that are shared between anxiety and schizophrenia, which are about 9,000. And so this means that genetic variants can be associated with multiple phenotypes, with multiple psychiatric disorders, which we saw at the beginning about the pleiotropy. And if we go on slide 44, we can talk about specific levels. So there are specific regions in the genomes that are shared between anxiety and other psychiatric disorders. So on this slide 44, we can see that anxiety, which we are talking right now of the plot on the left, which is in black, is connected with all these other psychiatric disorders that we are seeing here. So schizophrenia, PTSD, depression, bipolar disorder, ADHD. And so, for example, we see because of the high genetic correlation that we saw that there are a lot of regions that are shared with depression. Some regions with schizophrenia, some regions are connected with PST. And then we have again the DRD2 locus, which is about dopamine. These locus appear to be involved in different psychiatric disorders. As we saw anxiety, but again, like as I mentioned, schizophrenia, and then there is also depression, bipolar disorder, ADHD, and PTSD. And the effect of the variance in this region, in this gene, appears to be correlated across different psychiatric disorders. And so, which means that this gene is particularly important not only for anxiety, but also for other mental illness. And then, like, let's talk about other disease. So in slide 45, we did a phenome-wide. So the phenome is the collection of all human traits. And so we use information about more than 10,000 phenotypes to understand the connection between anxiety and other health outcomes. Of course, so here we can see on slide 45 the genetic correlation, again, RG, and the minus log 10p on the y-axis is the strength of the association. Higher the point, stronger the association. Each point here is a phenotype. And so like we have different colors for different categories. We can see that most of the strongest ones are reddish, which are related to psychiatric disorders, but there are also other traits. In particular, when we look at this, we saw that there were a lot of like pain, a related phenotype that had a high genetic correlation with anxiety. There were also several gastrointestinal phenotypes that had high genetic correlation with anxiety. And so like this means that probably that the genetic variants that are predisposing to anxiety are also predisposing to other disorders. Again, pain-related disorders, gastrointestinal disorders. And if we look to the people affected, we can see that people with anxiety tend to report like pain, chronic pain. They tend to have also gastrointestinal symptoms. And again, the analysis that we did showed that there is a genetic basis of these comorbidities. And then like I want to briefly, again, I probably talked a lot, much more than I was asked to. On slide 46, we did a brain imaging study. So in this study, we try to understand which were the brain regions that were kind of like more connected with anxiety. And we found two specific brain regions. These are not the only one. This is the one that we were able to identify because of the sample size that we had. And so like, so the two regions were the area of the right posterior middle singular gyrus and the gray matter per volume of the right anterior superior temporal gyrus. So these are brain regions. And so when we looked on what these brain regions do, if we go on slide 47, they were related to pain sensitivity. And if we look to the specific symptoms that they were associated with increased people reporting increased tense and sore and aching muscles during worst period of anxiety. So again, this is a connection that we see in a different study with different data about anxiety and pain sensitivity. And so like if we go on slide 48, again, few key messages. I talk about a lot of different things. Again, like genome-wide association studies are very powerful tools. So we can see here, again, and I didn't have time to talk about all the many things that we did in this study, about how integrating genetic information with other molecular data, multi-omics analysis, we were able to find new genes and mechanisms that help us to understand better what are the basis of anxiety predisposition. The other point that I want to highlight that this study is the most diverse that we have right now for anxiety, but is not enough. We see that there is still this overrepresentation of European descent participants with respect to the others. So currently, scientists around the world are collaborating with each other to try to involve more diverse populations. And again, like the other thing is that we need more molecular studies to try to understand the mechanism. So we found the gene, and again, you saw that we have different hypotheses, but again, like there are different mechanisms that can play a role. So this is not the final step, but is one of the first steps. We can say that this is not actually the first step, but is one of the big steps that move the field. We need many other of these steps before we can go back to patients and say, oh, again, I understand why you have the disease. I can help you because of certain things that we understand about the biology of the disease. We are not there yet, but this is, again, is a relatively big step. And so if we go on my final slide, 49, again, this was a group effort with a lot of people that contributed. Here I have my email, my social media, and again, this study was funded by the National Institute of Health, in particular the NIMH, National Institute of Mental Health, and One Mind. So I think I talked a lot, but I'm happy to take questions if people want to.
Thank you so much, Dr. Polimanti. Such a very, very thorough presentation, and we really appreciate that. It's some kind of awakening or information that we get from your study. So I would just like to reintroduce you to the audience for those who came in late. So our guest speaker, Dr. Renato Polimanti, is an associate professor of psychiatry and biomedical informatics and data science at the Yale School of Medicine. He is an associate professor of chronic disease epidemiology at the Yale School of Public Health. He is also an associate director for scientific and technical operations of the Clinical Epidemiology Research Center at the Veterans Affairs Connecticut Healthcare System. Dr. Polimanti's primary research interest is to apply big data analytics to molecular, clinical, and imaging data to understand the biology and epidemiology of human traits and disease, and to develop tools to improve the healthcare of diverse population groups. So I think earlier there was Nas who tried to come on stage to ask a question. So we have Kevin here, and we also invited Ade to the stage, but his question is here anyway on the room chat. So we will read that in a while. But I'm just very curious, Renato. The study that you have, everybody says that there's a rising, open close quote, epidemic of depression in the world, especially also among the people who are um, on their adolescent stage, also among students as well. What can you say about the relevance of the study in vis-a-vis this kind of situation that's happening to us?
So again, one thing is that we need to understand that there are different mechanisms contributing. Most of the times, the answers that we get from our study and from research in general is that things are more complex than we expect. So again, for several diseases, we are seeing an increase in prevalence. So there are more people that are being affected. And so like there are multiple things that play a role here. Again, it could be environmental factors, stress that can contribute. It's also another thing is that diagnostic criteria change over time. And so like there is, for example, the in the US, we are using the DSM criteria, which is the Diagnostic and Statistical Manual for defining psychiatric symptoms and classifying them in different disorders. And so, for example, the latest one is the DSM-5 that was released in 2013. And so, like, diagnostic criteria also changed. And so, like, changing in diagnostic criteria can increase the number of cases because clinicians are recognizing different symptoms are related to the disease that maybe like 20 or 30 years ago they were not kind of classifying them as pathologic. The other thing is also that awareness is increasing. So maybe like, again, 30, 40 years ago, people had symptoms, people were not feeling well, but there was more stigma about talking about it. And so people were kind of like not expressing their issues, uh, bottling up. And so like that could be also an issue. So there are multiple things that are going on that could contribute to this increase in prevalence. The environment, again, as I mentioned, diagnostic criteria that changed, and also like increased awareness. And so like what we can do, again, like the type of research that I'm doing is not something that will have an effect immediately. It's more about understanding like which are the biological mechanisms and how we can use this information to help people. So it's going to be something that takes a long time. Again, but the goal is to understand better which are the people that are predisposed to develop the disease, because maybe we can provide some preventive screening, uh, or also we can identify which are the genes, and then we can develop new drugs to target that specific gene. Because, for example, the drugs that we have right now were not developed because of like this large scale analysis. And so, like again, like this is the other thing that we hope to do with these large genetic studies, is developing new drugs that are going to be more effective to kind of prevent the disease or treat the symptoms. And sometimes treat symptoms that are shared across different disorders. But again, like human biology is very complex. Again, as I mentioned, like our genomes has more than 3 billion nucleotides. There are like more than 20,000 genes. And again, it's then differences there are between different cell types. So in our brain, there are many cell types, many brain regions, and it's very hard to understand which are the mechanisms that are predisposing to disease and what we can do to prevent them.
Thank you so much. Yeah, so my reaction also to the study that you presented, I think it gives people more understanding and maybe more interception to look at, the situation that we have now. I remember I was asked to endorse a milk commercial before, and one of the bylines that they were saying that if you take this milk, during your developmental period, is that you will become more gifted because it is trying to enhance nucleotides. But which nucleotides to enhance? They did not specify actually. So yeah, this is very important because I'm active in the gifted community, and I'm also doing this is like a hobby, but this is like advocacy work in the gifted community. So always the question with us is that what is giftedness? Is it nurture? Is it nature or nurture? And what can we do in order to increase the potential of these people or to fulfill the potential that they had when they were born? So I think if you have future studies along this line, please let me know. I would like to become your student on this aspect because it has been quite some time, and we've been talking to a lot of people who are studying clinical psychologists and also psychiatrists, developmental pediatricians, and yet I'm sure you know that we could not give a definite answer to this, but I think I will share your study with the other professionals who are also on this field. It will enlighten a lot of people. And yeah, I think you are right that criteria, and because of the new information that and the technology that we have are changing, because there's an article that I saw about autism and Asperger's, and in the past there is a subclassification of Asperger's, although it's a part of autism, that these are really highly functioning people. But I think in the DSM-5 now they have relegated Asperger's back because it is also so complicated that they could not look at the specificities of this one, and they need to study it more. So I'm very very curious about all of this because I think through your study, if it will be explained and expanded more, many people, it will change science maybe, and the way try to people try to look at heritability. And just one last question, I will yield the mic to the audience. So if we have people are also misdiagnosed, if for example they have Asperger's syndrome, it usually has a comorbidity with schizophrenia. So during childhood, Asperger's syndrome and schizophrenia are not diagnosed properly. What can you say about that?
So again, like the other issue about psychiatry, which is again is a challenge when we compare with other medical specialty, is that in psychiatry we don't have like biomarkers. So for example, for diabetes, again, the first test that people do is to check the glucose level that you have, and then there are other blood tests that people can do to refine the diagnosis. And so, like, these are objective and they don't change. Again, they can change because like parameters can change and technology can advance. But in psychiatry, we don't have that. It's more about the symptoms that people are reporting, and the training of the clinician to distinguish these symptoms. And so again, and as you mentioned, in children where you have a developing brain, things can change and they can change fast. And so again, a diagnosis can change. For example, both the two disorders that you mentioned about Asperger, which is now in the autism spectrum disorder, and schizophrenia, which is also a spectrum. Again, they are spectrum. So there are some symptoms that may be shared between these two and others that may be shared with other psychiatric disorders. And so again, a clinician will need to make a diagnosis based on the symptoms that a child and then a teenager and then an adult will have, and these symptoms can change because again, also psychiatric disorders are boxes where we are putting people with similar symptoms, and so it's kind of a modeling the clinicians have to do to kind of provide feedback and healthcare to patients. But again, again, in biology is never about different boxes where things are. It's always about a continuum. So we have a psychopathology spectrum, which is a continuum, and in this classification, there are certain things that overlap. So again, for example, psychotic symptoms, manic symptoms, depressive symptoms, internalizing symptoms, and all these things in some cases tend to overlap. And so sometimes there is not a clear diagnosis. And so my recommendations is to rely on clinicians that have the expertise. And so having someone that went through the right training. So for example, for a child that has some symptoms that are affecting their quality of life, the recommendation is to go to a child psychiatrist that is trained to recognize these developing symptoms. Because again, in children, adolescents, you have a developing brain, so symptoms will change. And so you need someone that has the expertise to kind of understand which could be the trajectory. And this kind of symptom trajectory can change. And so it can be very hard to predict what will be the course. And so again, unfortunately, I don't have most of the times I don't have a simple answer because we don't have simple answers for complex problems. But it's about relying to people that want to extensive clinical training to recognize this and kind of trajectory of symptoms that people will have. Because again, sometimes they may reflect symptoms that are related to different disorders. And so understanding which is going to be the disorder and which could be the right treatment can be very hard. And so again, rely on clinicians, find a clinician that you trust, and again, it's a very important point.
Yeah, thank you so much. And now we'd like to give the mic to the people here on stage and also check the questions that are on the room chat. So we usually try to give the speakers here on stage the opportunity to speak based upon PTR order. But this time, I invited Ade. I hope I pronounced your name correctly earlier, but maybe it's only now that Ade is able to come up because during the start of your talk, Ade had already typed in his message at the room chat. So this time I'd like to give the mic to Ade, then get to Kevin, and after that to Jonathan. So Ade, if you would like to speak now, please unmic.
Okay, thank you, Cecile. But my English is not very good for speaking. I will write in the column chat for my question to the doctor. Thank you.
All right. So Ade, while you write your question, which Bob will read in a while, I'd like to give the mic to Kevin now.
Okay. Yeah, my main question is about epigenetics, and so do you think epigenetic mechanisms, contribute to the development of anxiety disorders, and are there plans to investigate epigenetic changes in future studies, and the potential for the role of gene environment interactions in anxiety disorders, and how epigenetic mechanisms might influence these interactions, and any plans to explore brain region specific epigenetic changes in anxiety disorders, and how might this knowledge inform the development of new treatments like personalized approach. So that's hopefully yeah. So that's basically my question.
Yeah, that's a very good question. So again, like we talked about different molecular mechanisms, again, how the genome is complicated. So for people that are not familiar about epigenetics, so epigenetics is another modification that can happen in the genome, and differently from genetic variation, which is fixed. So again, if we are not talking about somatic mutations that are more related to cancer, the genetic variation that we have is that one that we are born with, so it's not going to change. But then, like the genome can also regulate itself also through epigenetic mechanism. These are modifications that the genome can make on its nucleotides and on proteins that regulate the genome assembly. And so, like again, and this can be again kind of like regulated by genetic variation itself, but they can also be induced by environmental factors. So, for example, stress, being exposed to a stress or having a certain diet can play a role in modifying epigenetic variation. And this can induce gene expression changes, because again, modifying the epigenetic variation can induce changes in the regulation, and this can induce changes in the predisposition of anxiety. So again, like this is an area that people are kind of like expanding. Again, like the first GWAS was done in 2007 or 2006, and the first epigenome-wide studies came many years after. I think at least 2010, if not later. But again, like the challenge is also that there are different mechanisms that can link epigenetic variation to a disease. Can be the epigenetic variation that induce an increase in the disease, but also the disease itself can induce changes in epigenetics. Instead, with genetics, we have only one direction. It goes from the gene to the disease. And so, like, the goal right now is to expand the sample size of epigenetic studies. For example, I'm aware of a large epigenetic study that includes more than 40,000 participants, and you can see how different it is. So here I'm talking about a genetic study of more than 1 million individuals, which kind of is the standard right now. But for epigenetic research, we are still growing. And so 40,000 is a massive sample size for epigenetic research. The other issue is about like that genetic variation is the same in all the cells of our body. Is it the same in the blood? Is it the same in the brain? And so, like, if we collect blood and analyze the genetic variation, we can use that to gain information about different biological systems: brain, heart, gastrointestinal system, and so on. Epigenetics is not like that. Epigenetic variation that we have in our blood cells is going to be different from the epigenetic variation that we have in the brain. And so, like, unfortunately, to collect brain samples, we need to do it on people that died for different reasons. And so the sample size of 40,000 that I mentioned just now is about an anxiety study done on blood samples. And so, like, if we talk about anxiety or psychiatric research done in brain, then we go from 1 million genetics, 40 million blood epigenetics, few hundred, probably 1,000 for brain epigenetics. And so this means that, again, the less samples we have, the less power we have to investigate the complexity. But the goal is to have all these studies done in parallel, and each of them will provide more information about all these different molecular mechanism, genetic, epigenetics, transcriptomics, metabolomics. I saw someone in the chat mentioning microbiome. And so again, all these things will need to grow together, and like pieces of the same puzzle will grow together, and at certain points they will converge and they will inform each other. So that's the hope. The more data that we are generating, right now, most of the things that we are generating may not give us a complete picture, but the more data we are generating, the more we are understanding the complexity of the disease, and at a certain point, we expect that these things will converge and will give us a more clear picture. So, but again, like epigenetics is going also to be very important to understand the impact of the environment, because the way that the environment is acting on biological systems is probably through epigenetic changes and the other like molecular changes. And so like we need to understand that. So it's going to be very interesting. Again, it's still very interesting, but again, the more data we are generating, the more it's going to be interesting to understand how all these things come together.
Oh, and maybe, well, that makes me think of more speculative, you know, for the new year, you know, with all the data, you know, do you think quantum computing models, you know, coming out soon, you know, statistics, forecasts, and new drugs can be helpful for this, or, you know, just to speculate, I guess?
No, again, like, this is another great point. So again, like one of the key messages that I wanted to give people is that things are complicated. We have complex biological systems that act one on top of the other, that interact with each other. And so again, all these data that we are generating are already requiring like high performance computing clusters to be analyzed. And again, the goal is to develop more sophisticated, again, because what we do to kind of like generate data, the information from this data is to define models. And of course, the models are more simple than the real thing. And so like we need to have more complex models. And so for example, AI, machine learning, there are people that are working on different research areas, like for example, computational psychiatry, to kind of model which are the molecular mechanisms that happen in the brain. And so the type of research that I do can inform this kind of research. And again, like the goal is to have more data, but also more complex modeling that can understand better how all these different biological processes act together in different conditions. Again, we talked about before about how the symptoms change between children, teenagers, adults. Why? Because the biological mechanism in the brain and also in other organs are changing. And so like it's a dynamic thing that we are analyzing. And so for example, in the study that we had a single time point. We asked about anxiety in their life. But again, like we have trajectory, symptoms that play together. And so like it means that we need more complicated modeling to understand that.
Thank you, Renato, and thank you, Kevin. So, yeah, I'd just like to read some comments here of Ade because he's supposed to be next. And it's up to our speaker to explain some of the things because he's talking here about something. Yeah. So, Mr. Renato, can I ask something about schizophrenia? Please explain from a biological point of view what causes schizophrenia. And there's a follow-up comment. He said that I am a 10-year schizophrenia survivor, and in my independent research of his illness, he concluded that dopamine is not the only cause of schizophrenia, but also inner emptiness and inner damage. Is there a logical explanation for the inner biology or any other science? What I mean is that it does not only happen because of brain damage, but also because of ongoing damage in the soul or conscience. And I think, yeah, that's all that he wrote here. But before I give the mic to you, Dr. Polimanti, I want to ask Ade, uh,
Yes.
how and who diagnosed you to have schizophrenia?
The diagnosis. how can I can explain that? Wait a minute. The story is like this. I have wait a minute. How to? I took the medicine to control dopamine in one day. 200 milligram per day. In 7 or 8 years, but hallucination still there. I think I don't know the hospital diagnosis me schizo because I have full emotion after the tragedy like that, Cecil. I mean,
Maybe
I cannot.
PTSD, I
Mm,
think, yeah, post-traumatic,
mm,
yeah, syndrome. So
yeah. Mm.
anyway, I was just very curious because how were they able to diagnose you that you really have schizophrenia, and was there a professional doctor who diagnosed you for that? And I speak to that because I'm curious about the medication that you have and
Mm.
also your description about conscience. So I was wondering. So I'd like to pass. Sorry, doctor. I'm just very curious. I'd like to pass the mic to Dr. Polimanti.
So again, like that's a very good point. Again, like because I mentioned in my talk about the RD2, which is the gene that encodes for a subunit of the dopamine receptor. It's a very good point because again, like schizophrenia is not caused only by dopamine. And again, as I mentioned, like the RD2 is one of the variant that is in the RD2 that is associated with schizophrenia, but we have also associated with anxiety. Is a variant with a very small effect. So dopamine is one of the many mechanisms that probably play a role in the predisposition to schizophrenia. There are different hypotheses. For example, one of the strongest association in schizophrenia is in the major histocompatibility complex, the MHC region, which is a region that plays an important role in the immune system. And a study from a few years ago, I think almost 10 now, showed that the effect of these variants that have a relatively large effect size, but not large as the rare variants that I showed you before. Among the top variants in the common, so the small blue ones, like if we, let me go back to the slide, so if people want, they can check it on the slide. So if you go on slide 15, there is this plot where there are rare variants, which have a very low minor allele frequency, and common variants. C4 is the gene that I'm mentioning. So you can see that the effect is, compared to the other ones that are rare, is very small, but it's the top and so the mechanism that is underlining that association is about synaptic pruning. So synapses are the connection between the neurons, and so during our childhood there is a synaptic pruning. So some synapses get kind of removed because they are not needed. And so like the mechanism that should be underlying C4 is that the variant that we see associated with schizophrenia in C4 increase the synaptic pruning. So more synapses are removed from the brain that they are supposed to. And this could be one of the mechanisms that is predisposing to schizophrenia. But again, like, because there are so many variants, right now we people identified more than 270 variants associated with schizophrenia, and probably there are between 5,000 and 7,000 variants underlying schizophrenia predisposition. And so it means that all these variants, again, not each one of them, it's not that there are 5,000 different ways to get schizophrenia, is that some of these variants group with a specific mechanism that may predispose certain individuals to develop this disease. In some cases could be dopamine, in some cases could be this synaptic pruning that I mentioned. And again, the other thing is also like how we connect biological mechanism with feelings that people are having, symptoms that people are expressing. Again, the person in the audience mentioned about emptiness. And so again, the thing is that one of the goals of scientists right now is to move from case control analysis, again, analysis where we compare people with the disease to people without the disease, and looking at people that have different symptoms. So people that are reporting different feelings. Again, like for example, for schizophrenia, people that have hallucination, like visual hallucination, auditory hallucination. And so trying to understand how all these things work together. And so in this way, we can understand better which are the biological mechanisms that are predisposing to these different symptoms, different feelings, and if there are drugs that we can find that target these different genes to prevent these, again, pathologic symptoms.
Thank you so much. So, Ade, I hope Dr. Polimanti has answered your question. So,
Okay,
yeah,
thank you.
thank you. And we'd like to pass the mic to Jonathan.
Thank you, Cecile. Dr. Renato, first question is, I was astonished to see that the number of GWAS, the size of the GWAS sample is up to 800 million currently?
So these are all the studies conducted. As I mentioned, there are more than 7,000 studies, and so samples were collected around the world. And so for individual studies, like I think the largest study, individual study, includes about 5 million individuals. But if you put all the samples that were genotyped around the world, that's the sample size that we have right now, which again is massive, but is increasing very, very rapidly. Like again, there are many more that are coming up.
So that 800 million number represents genome-wide full sequencing, or what does GWAS mean in this case?
Yes. So let me explain. So again, we can genotype, so we can understand the genetic variation of individuals with different kind of technologies. So early on, when again the technology was very early in its infancy, like again we were doing genotyping each variant individual. Then people developed some sequencing technology, and so from single variants we were able to sequence, kind of analyze an entire piece of the genome. And then, for example, there was the Human Genome Project, where the first full genome was sequenced in the 2000. And then from that, people started to develop arrays. So arrays are the one that we are now using for genome-wide association studies, where in an array, we already know which one are the positions where people have variants. And so in this array, there are probes for each of the variants that we know. And right now there are different methods that we can use to convert an array that have information about 500,000 variants to have like information about.
Okay. So this is not a complete sequence analysis.
No. Like
individuals.
the whole genome sequencing analysis are increasing because again, like.
Right. Okay. Actually, I worked at the Howard Hughes Medical Institute Genome Mapping Library Project at Yale for about 6 months, a long time ago. So I know some of this history. But thank you. Okay. So GWAS is a sampling of the genetic diversity.
Yeah.
Okay. Got it. Thank you.
But
okay.
Again, like in few years, we may have like that. Most of these people will, again, in addition to having GWAS, like genome-wide genotyping with arrays, we will generate whole genome sequencing. Again, the issue is always the cost. Again, right now, like until a few years ago, to do a whole genome sequencing analysis of a person, it would cost like $1,000 or euros. Again, if we think how much costed the first whole genome sequencing in the early 2000s for the Human Genome Project, which was in the billions, again, the cost went down. And it's still going down. Now we should get high quality whole genome sequencing for a few $100s. And so again, like the prices are going down, so it means that scientists can analyze larger sample size. And so hopefully we will move from GWAS to whole genome sequencing studies, where we are going to analyze not only common variants, but also rare variants. And this will help us to understand better how the predisposition to complex traits work.
Okay, thank you for the clarification and bringing me back up to date on that. So the second question you picked up on already when you indicated that you're going to be expanding into microbiomics. And so seeing that the there were a couple of gastrointestinal variants that were associated in slide, I think it was 46 or 47, with the psychiatric diagnoses was fascinating in terms of gut microbiome communication with the brain. So that's an area of active research in your lab or
So
associated labs?
yeah, so like we have some data about microbiome, but again, and again, it's very interesting, but it also is also very complicated. So the thing that I want to clarify is because again, scientists, but also people get excited when there is a new technology. So when the genetics came in, and because usually I have a slide about the hype and the crash when so when genetics came in, for example, when the first whole genome sequencing of the Human Genome Project, people say, "Oh, we sequence the entire genome, and so we know which are the genes, and we will know how these predispose to disease." And then we discover that things are more complicated. It's not a single gene, but there are multiple genes. And then we add the epigenetics. So now we are studying epigenetics. And so the solution is not going to be genetics because it's too complicated, it's going to be epigenetics. But the more we study epigenetics, the more things are getting complicated. And so that's the thing. And then came microbiome. And again, the solution is microbiome. It's not genetics, it's not epigenetics. But again, the more we study the microbiome, the more we see that things are complicated. So again, the thing is that all these different pieces are contributed to something, but they alone cannot explain the complexity of human biology. And so, for example, again, in this case, for anxiety and gastrointestinal disease, for sure, understanding better how the brain and the microbiome interact is going to be very interesting, especially for understanding about gastrointestinal symptoms in people that have anxiety, and also how environmental factors play a role, how the diet of people with anxiety play a role. Because again, people with anxiety may have predispositions to have a certain type of diet with respect to people that don't have the disease. They may have a different predisposition to enjoy physical activity or not enjoying physical activity. May have a different predisposition to drink alcohol or not drinking alcohol, to smoke tobacco, not smoke tobacco. And so you can see how all these variables work together. And again, it takes a lot of work to try to understand which are the mechanism. Again, we need to create a model that can integrate all this information at the same times, but not only in a single time point, but longitudinally, like how these things change over time. And so again, it's that's how it's completed. Again, microbiome is going to be very useful, but it's not going to be the solution by itself. It's going
totally,
to
totally understood. And I want to, if I could, ask just one more question. And I'll try to keep it short. But you had a very good segue into my question when you were talking about the behavioral predispositions of people with different psychiatric diagnoses. So, as a former evolutionary biologist, also at Yale, hello, New Haven. Um, I'm very interested in the uh, possible evolutionary psychology perspective in terms of perhaps these conditions that are currently called disorders may have in the past, when our population sizes were very small, below the Dunbar number of 150, of possibly having been maintained by balanced polymorphisms and the adaptive value of the different psychiatric types in terms of the fitness of the group with respect to dealing with threats, with different functions within a tribe, such as having individuals that are more prone to anxiety, being kind of the watch people of a tribe, or individuals that are, um, um, perhaps would be characterized by anthropologists as having been the oh, what's the term? Um, the spiritual leaders, the visionaries,
Yeah.
Was there perhaps from a legitimate from your perspective as a currently practicing um, genomic scientist, do those sorts of perspectives of these conditions not being disorders but possibly having been maintained by balanced polymorphisms in the past, is that at all a credible perspective, or is that more of a pop psychology romanticization?
So again, like that's something. For this summer, I went to a workshop when I discussed it with other people from other fields about evolutionary hypothesis to explain like psychiatric disorders. And again, like there are multiple things here going on. So let me start to answer to this question. First of all, we need to understand like that again, like one of the things I mentioned is that psychopathology is within a spectrum, but also like each of these symptoms, like each of anxiety symptoms, psychotic symptoms, autistic symptoms, they are also in a spectrum. And so like there are at the end of the spectrum people that have like kind of like very severe symptoms, and so like that they are developing the disease, because again the disease, what is a disease? The disease is a state where people are having issues to manage their life. And again,
Mm-hmm.
like for example, there may be people that are high functional that may have some anxiety symptoms, but they may be highly functional. And so if we think about these individuals in an environment like the environment that have early human beings, where they had like predators that may attack them and they have to hunt. And so like being that reactive, being like that ready to flee or ready to hunt could have been an advantage. And so like we may think about that these feelings that people have may have been useful when they were on the physiological side. And then like people that developed this extreme one were kind of like the so in the population it was good to have like people with some of these mild symptoms, but then we also had some symptoms that were very severe. And so like it's something that people are still discussing with respect to genetics, because again like one thing that we can do is also analyze uh, like the because again the human genome, it's an amazing thing, because again can explain us about the predisposition to develop certain physiological or pathological outcomes, but they can also includes the signature of our past. We have the signature of our demographic history. The genetic variation that we see among human
Mm-hmm.
populations is primarily driven by migrations that populations had. And also we have adaptation processes. So we can see signature about genes that were useful to adapt to different environments, to react to different pathogens. Again, for example, the immune systems in humans were strongly shaped by the pathogens that we were exposed to. With respect to psychiatric disorders, because of the high polygenicity that we are seeing, so with respect to other disorders, we see that for psychiatry outcomes, we have more variants with smaller effect size. And so the primary evolutionary pressure that we see there is the purifying selection. So we see that because the
Mm.
human brain is so unique, is so complex, it doesn't allow to big changes. So like when we have like a new mutation that happens randomly, and this new mutation is affecting the brain, if the effect is large, it's going to be most likely deleterious, and so the mutation is going to be removed
Mm.
from the population. So that's what purifying or background selection is. And so this is why we have all these variants with very tiny effect size that are associated with psychiatric disorder. And so like again, like right now geneticists are still trying to understand how we can model orogenic signatures underlying psychiatric disorder to see if there are adaptation processes. Because so far, the only thing that we are seeing is this strong purifying selection because of the complexity of the human brain. But probably there are other things going on, so it will require more research. And so like there may be adaptation underlying secondary disorders, but so far we don't see that from the genetics because we see this strong purifying selection. But again, like as I mentioned, like the more we know, the more we understand better center dynamics. And so there are people that are working really hard. Again, it's a topic that is I'm very interested in. And so like I'm working also with other people to try to understand these dynamics. But so far in the genetics of psychiatric disorder, we mostly see this purifying selection, which also is active on other diseases. But it seems that it's acting more strongly on psychiatry because of the brain.
And this is a subset of just very, very quickly here, because I realize it's getting late. The average population sizes of humans in prehistory, before, let's say, 5000 BC, was often very small of a breeding group, and often there were very severe bottlenecks and founder effects. So I imagine purifying selection would have been especially rigorous in such times.
Yeah. Yeah. So like again, like the other challenge about human diversity, because it reflects all these bottlenecks. For example, one of the first bottlenecks that shaped the genetic diversity of human population is the one that is called out of Africa. So human species is an African species. So the first anatomically modern humans were in Africa. And so like within the African continent, we have more genetic variation than outside the African continent because of this bottleneck. So a subset of humans went out of Africa probably multiple times. And so that generated the genetic variation that we see outside Africa. So there is, again, more variation inside Africa than outside Africa. And so this is one of the first challenges because again, like African population and African descent population are going to be more informative for their diversity. And so again, this is one of the most interesting thing about genetics because everything is connected. The things that shaped in the past, the genetic variation of human populations are going to be having like a role in also like shaping the genetic predisposition to certain disease. So for example, in some cases, we may see that some disease may affect certain population more than others. This is not the case, for example, of psychiatric disorders, because again of the higher polygenic architecture, but for example, like African American men tend to have a higher risk of prostate cancer because of one of these genetic adaptation that happened in Africa. There are other mechanisms that happen like in East Asia. For example, there are more people in East Asia that are highly reactive to alcohol drinking. There are some people that when they drink, they get sick immediately. And for example, there is a high percentage of these people in East Asia. And so again, all these dynamics also play a role. So again, as geneticists, we need to be trained both in the evolutionary side, the population side, but also being able to connect these on the disease that we are interested in.
Thank you so much, Dr. Polimanti. An excellent presentation and really profoundly impressive research program you have. Thank you.
Thank you for the nice questions.
Yeah, thank you so much, Renato. Yeah, I promised you that I will ask you this question at 1.5 hours time. But we've been here for already 2 hours, and I think that it's getting late on your side. Although we have 1,000 questions, and I think there are 3 questions that was sent to me. So yeah, I would like to thank you very much for sharing your knowledge and your time with us, and we hope that we can invite you back again here in Quantum Photonics.
Of course, again, it was a pleasure. Again, I enjoyed so much both talking about my research, but also like answering questions. They were great questions, very, very interesting. And so again, I'm happy to come back to talk about other topics. And people can feel free to reach out to me via email or via social media. I'm happy to engage to people again. I think it's very important for people to know about what scientists are doing again. And also when in general, when you read about research that you may be interested in, or again, you may want to know more, I invite people to contact the scientists, because again, one thing the scientists are becoming more aware of is to be able to communicate with people that work in other areas, and so like to explain what we do, because again, most of the research that we are doing is funded with taxpayer money, and so like we need to be back to the community, not only in terms of like knowledge, scientific knowledge, but also back in terms of information about what we are doing.
Yeah, thank you so much. We are glad that the mission of Quantum Photonics align very much with what you have in your heart, because basically that's why we bring
Yeah.
serious and excellent researchers like you here in Clubhouse. It's because we want science to be nearer to the people, and we would like the general audience to see how wonderful science is, and how also very helpful it is to societies. So yeah, with that, thank you so much.
Thank
I
you
will
again.
the papers that you have, and I will invite you again soon. And you are so generous that it is with great appreciation that we are going to close this event now. So I'd like to give the mic to Bob, who might have some messages.
I wanted to express appreciation for your slides, which are packed with information and laid out eloquently. You've set the bar pretty high. Thank you, sir.
Thank you again. Again, it was a pleasure, like to prepare the slides and also like to show them to everyone.
Yes, so let's flash our mic for our great speaker. Yeah, so thank you so much. Thank you, Kevin, Ade, and Jonathan. So let's give the mic to Darcel to help us do the countdown to close the room. And to everyone in the audience, see you again in the next few days. Please check out our upcoming rooms in Quantum Photonics. And thank you, Dr. Polimanti, and more power to the Poliman team.
Thank you. Have
So,
a good night.
yeah, good night and happy Thanksgiving to all.
See you
Uh,
Thanksgiving.
Mike, you there.
All right. Well, thank you for a great room, Doctor. To close the room, we're gonna start our countdown from lucky number 7. Is everybody ready?
Seven,
All right, here we go. 7,
seven,
6,
6.
6,
Jonathan 5,
5,
Eddie 4,
4,
Kevin 3,
3.
Pato Saques.
2 for me, please.
So, we're gonna have to speak for our guest.
Danke.
All right.
Numero uno.
Numero uno. So, yeah, good night, everyone. Happy Thanksgiving and see you next week.